Hello everyone. Welcome to the webinar on gene editing entitled Food Futures, Commercialization of Gene Edited Crops in Asia and Australia, organized by ISA, Murdoch University, and PACE, Package Assisting Small Exporters. We also acknowledge the help of our network of biotechnology information centers in disseminating the information about the webinar. Currently, we have around more than 100 participants, and others are still joining in. I am Panfilo de Guzman from ISA Asia Center, and I will provide a brief overview of what to expect from the webinar, as well as some house rules. Recent developments in the regulatory status of gene-edited crops across the world are opening up significant commercial opportunities for agribusiness. The sustainability of food value chains depends on a robust science, policy, society interface. This webinar will bring together important scientific and regulatory updates in Asia and Australia that relate to the future of gene-edited plant products. Experts from the academia and industry in Asia and Australia will provide significant science regulatory advice for relevant stakeholders to enable trade in produce developed using gene editing technology. Before we start, a few house rules. This webinar will run for 2 hours and will be recorded for documentation purposes. The webinar will also be streamed live on ISA's Facebook page, and the recording will also be made available later on ISA's YouTube account. Make sure that you have good internet connection so you won't miss any of the discussions. Your camera and microphone will be turned off during the sessions to avoid any distractions. We encourage interaction and exchange. Thus, we welcome questions from the participants. Please use the Q&A button at the bottom of your screen for any questions and clarifications you may have. You may also click the raise hand button if you want to speak, and we will unmute you. We will entertain the questions during the open discussion. Earlier, we asked you to answer a pre-webinar survey, basically intended to gauge your level of understanding, knowledge, and appreciation of new breeding technologies. Thank you for participating in the survey. We will again ask you to answer a post-webinar survey at the end of the webinar. This will be the basis for the issuance of your certificate of participation. Thank you very much for taking your time in attending this webinar, and we look forward to your active participation. Good day to all, and I will now pass the screen to Adil for the first session. Adil, please.
Thank you. I hope I'm audible to all.
Yes.
Yeah. So it's a pleasure to be moderating this first session. So without wasting further time, I'll just get right into it. So the first session is on the research, policy approaches, and implications for market and trade. So country perspective. We'll be hearing country perspectives from experts from China, Japan, India, and Pakistan. So first, our expert is from China, Dr. Wei Zun. She's affiliated with the University of Science and Technology in Beijing. Her area of specialization include agriculture economic management, experienced project manager in agriculture science and technology programs belonging to the Ministry of Science and Technology. Her current focus is on tracking the trend, resource allocation, and commercialization of policy environment of biotech and other cutting-edge agriculture technologies. Dr. Wei Zun, you have the floor. Thank you.
Thank you. Good afternoon, everyone. And I'm Wei Xun from University of Science and Technology, Beijing. And I'm so honored to be invited. And today I will talk about something about the policy environment and commercial policy of commercial potential of gene-edited crop in China. Today, I will focus on 3 aspects. First is current policy system. The second one is main innovative achievement and the commercial status. The last one is industry collaboration potential. First, let's talk about something about current policy system in China. In general, all crops and their products obtained through gene editing technology are treated as agriculture GMO in China. Agriculture GMO is defined in China as animals, plants, microorganisms, and their products that use genetic engineering technology to change the genome composition for agricultural production or processing. So the gene-edited crops are definitely included. In a word, policy on gene-edited crop equals to policy on GMO. And the system consists of three parts: the supervision policy, the innovation policy, and commercial policy. First, let's see the regulations. The main regulations consist of 4 documents. The first one is regulations on the safety management of agricultural GMOs, which is issued by the State Council, and in 2001, the other three are issued by the Ministry of Agriculture, and separately in 2002 and 2006. These 4 documents covered the main requirements of safety evaluation, the import safety, and the marks. In recent years, safety evaluation guidelines and a series of testing standards have been also formulated. When it comes to the authorities, it has 2 parts. The administrative institutes from the top to the bottom is joint meeting of relative ministries, the Biosafety Management Office in Ministry of Agriculture, the Provincial Biosafety Management Office, and at last, it is required that every institute related to the biotechnology research and applied needs to have an internal biosafety management group. On the other side, there are some technology supportive institutes such as Agricultural GMOs Biosafety Committee, Standardization Technical Committee, and some testing organizations, all of which are managed by the Science and Technology Development Center, MOA. Yeah, it is. This slide is about the management process. It is the biotechnology from the lab to the customer. First of all, it has to be through the safety evaluation. With the safety certificate, it can through the next step, which is crop variety registration. If it got a variety certificate, it can be produced and do some business. And at the end, it can go to the processing link with the processing permit and then to the customer. In a word, whether it is safe or not is determined by scientific evaluation. Whether it can be planted or not is handled according to laws and regulations, and whether to eat or not is chosen by the customers. And here is the 5 parts about the biosafety evaluation chain, which is the laboratory study, restricted field testing, enlarged field testing, productive field testing, and apply for the biosafety certificate. In the last slides, I talk about the crop variety registration, but there is no approved variety yet. The measure for examination and approval of major crop varieties stipulates that the measures for the examination and approval of GMO varieties shall be formulated separately, but so far, the specific rules have not been issued. So one step before the crop variety registration is safety certificate. In 2009, there are 2 cans of rice and 1 can of maize got their safety certificate, but due to the public concern, the certificate has suspended for 10 years until 2019. When it comes to the import approval chain, it can be classified by purpose and by applicants. If a domestic institute want to import the materials for research and experiment, the laboratory study and field test is needed. If an overseas company want to use the materials for production, the field test is needed. And if the overseas company want to use it as processing raw materials, the biosafety certificate is needed. Here is the detection index for import as processing raw materials. We take transgenic herbicide-resistant crops as example. Yeah, the identification confirm must be done to identify the molecular characteristics. and the survival competitiveness, the gene drift, the impact on biodiversity has to be reviewed for ensurement of environment safety, and the anti-nutritional factor and the 90-day rat feeding tests have been done to ensure the food safety. Here is something about the R&D investment policy. All the green ones are the national key project or special project related to the biotechnology research and development. And all the blue ones are about the plan, both the short-term and the medium and long-term plan related to the biotechnology. You can see the first national program can date back to the 1986, which is National High Technology Research and Development Project. And in the recent plan, which is in 2020, the Central Economic Conference, which is very important in China and often held at the end of the year, it is said that respect science, strictly supervise and orderly promote the industrialization of biological breeding, which is believed a very positive signal to the commercialization of biotechnology. The second part, we will review the main innovative achievements and the commercialization status. Here is the research papers and the core patent of main countries. We can see China ranked the second according to the amount of research papers and core patent, but we believe that the quality but not quantity is more important and have to be enhanced. Here is the production approval. Among the 8 kinds of crops got the safety certificates, such as the tomato, cotton, the papaya, the corn and rice. Only BT cotton and disease-resistant papaya have been commercially produced, and all the other crops still stopped at the safety certificate phase. And here are some raw materials who got the import approval. It covers 6 crops and have 59 raw materials. Here is some GM foods you can see in the China market, including cottonseed oil, the papaya, the soybeans, corn, the rapeseed oil, and syrup, and other some relevant foods added with genetically modified food additives. And here is the 4 main agriculture companies. They have been laid out the industrialization work. For example, the Syngenta, which is acquired by the Sinochem in 2017, has reached a global license agreement with Broad Institute on CRISPR-Cas9 gene editing technology, and it is also applied to research and development of corn, wheat, tomato, rice, sunflower, and other crops. At last, let's see the near future of gene-edited crop market. Gene editing technology has shown as it advances in efficiency, accuracy, and economy. In the future, we believe a large number of new varieties of gene editing crops will appear, and the market capacity will continue to increase. According to the markets report, CRISPR plant breeding market is expected to reach $14.6 billion by 2023. And the Asia Pacific region is expected to have the highest increase. According to the report of the World Economic Forum, by 2030, 10 to 15% of farms will choose to use genetically edited seeds, and definitely it will bring some obvious positive impact, such as income increase, yield increase, food loss reduction, and macronutrient reduction. Here is the management framework expectations from China aspect. And we believe the management method of gene editing products contain foreign genes should be consistent with that of traditional GMOs, but Gene editing products that do not contain foreign genes can be supervised with loose but clear and accurate regulations. Here, the last one is the industrial chain layout expectation. We divide the chain into upper reach, middle reach, and lower reach. The green one is already we have some institutions and some suppliers, but the green one is still the blank. Yeah, for example, we had Chinese Academy of Science, the Peking University as a main university and research institutions, especially doing a leading work in CRISPR. They can provide productions to product suppliers such as a gene company, gene tech company, and so on. And they also provide service to the technology development service provider. And the product supplier can provide the kit, the genome-wide knockout screen, and other technology to the breeding company. And the technology development service provider can provide patent authorization and technology development to the breeding company. But unfortunately, there are still the blank of the market. Yeah, that's all. Thank you for your attention.
Thank you, Dr. Wizun, for this important and a very clarity-driven presentation. Next, we'll move on to country perspective from Japan, where we have Dr. Meiko Kasai. She is a professor, of Center for Environment, Health, and Field Science, Chiba University. Chiba University has the largest scale plant factory facility for research and educational purposes in Japan, and accepts many international students who come to learn the latest technology on indoor agriculture. She is also responsible for internship program for those students. She's a part-time lecturer of College of Humanities and Sciences, Nihon University, Department of Japan Plant Factory Associations, and serves as a biotech affairs consultant. Prior to working at Chiba University, she worked for DuPont. She has over 25 years of experience in biotech regulatory affairs and has contributed in development of science-based regulatory policies on genetically modified organisms and gene editing in Japan through actively participating in industry initiatives such as International Life Science Institute of Japan, Japan Bioindustry Association, Global Industry Coalition, and CropLife, etc. She's been participating in the Convention of Biodiversity as an industry member since 2008. She received a PhD at North Carolina State University, USA. Doctor, you have the floor. Thank you.
Thank you very much, Dr. Adele, for the kind introduction. Can you hear me okay?
Yes, absolutely.
Okay, thank you. Good afternoon, everyone. My name is Meiko Kasai of Chiba University. It's a pleasure to be here to introduce you the Japanese policy on genome editing. I'd like to thank the organizers for the invitation. As you know, Japan is not a GMO cultivation country due to consumers' concern, but as for genome editing, the Japanese government positioned the genome editing as one of the key innovative technologies which countries should invest. So in 2014, the government established the so-called SIP program, which is a strategic innovation promotion program, and identify 11 key areas to focus. And agriculture is one of those. And under this initiative, the government has started funding an R&D project with a goal of actually bringing the product developed through genome editing to the market. So this slide shows some example of the product under development. I won't go into detail for the sake of time, but some of the products such as Hijab Tomato, which I am explaining later, potato, rice, and tuna were developed under the SIP program. One thing I'd like to point out here is, as you may notice, most of the products have direct consumer benefit, and this is a Japanese government intention to do so to gain public acceptance of the technology. To bring the product developed through modern biotechnology like genome editing to the market, it is indispensable to have a science-based and applicable policy in place. So when the government started funding the R&D project in 2014, they also initiated the discussion on regulatory aspect. And after 5 years of discussion, the Japanese government has published the policy in 2019. So starting from the next slide, let me explain what it looks like, as well as what sort of consideration was given during the policy development discussion. Since GM technology is used at the early stage of genome editing, and also genome editing result a different type of product, whether or not you use template and what sort of template you use, the government considered each case separately and discussed what type of product are subject to the GMO regulation and what's are not. So here is a summary. For STN1 type product, which is a simple gene knockout without using template, the product was determined as non-GMO under the Food, Feed, and Environmental Safety Regulation, which is the Cartagena law. I listed each competent ministry, MHLW, MAFF, and MOE MAFF. For STN2 type product, which is a short template resulting the target mutation, and for STN3 type product, which is an introduction of new genetic materials. As you see, there is a discrepancy between the food, feed, and environmental safety regulations, and this difference comes from the different definition of GMO. So let me first explain the GMO definition under the Cartagena law. Its read, LMO is an organism possessing extracellularly processed nucleic acid, excluding the case that nucleic acid is from the same species or from sexually compatible species. So by applying this definition in a very strict manner, even though we are talking about a short template resulting just 1 to few base pair mutations, if those extracellularly processed nucleic acid are inserted into the crop, by definition of the law, such crop is GMO. However, the law also says if those nucleic acid are from the same species or from sexually compatible species, the product can be regarded as non-GMO. So this is a case-by-case determination. On the other hand, the definition of GMO of the food sanitation law and the feed safety law is the organisms obtained through recombinant DNA techniques, so that this is a process-based definition. But when you read the definition of recombinant plant, it said a host containing recombinant DNA. So this is a product-based definition, but there is no mention of origin of DNA. Consequently, the food sanitation law and the feed safety law doesn't give a clear-cut guidance of which genome-edited product should be a subject to the GMO regulation. So the approach they took was to make the determination based on whether or not the genome-edited product can be distinguished from the product obtained through spontaneous mutation or mutation breeding, from sequence change point of view as well as the food safety point of view. To make that determination, the 3 points shown in this table were considered. Deletion and change of 1 to few base pair happen, off-target mutation happen, and selection and backcross process apply. To the first point, the answer is yes for both genome editing and conventional breeding. To the second and third point, the answer is also yes for both methods. So this means it is difficult, if not impossible, to distinguish genome-edited product from conventional breeding product. Regarding the off-target change, even if the off-target change can happen with genome editing, many scientific papers show its frequency is within the range of undesired mutation caused by mutation breeding, and thus the risk is considered very low. So, and on the top of that, we all know that a long history of safe breeding has proven that those unintended and undesired change are eliminated during the selection and backcross process. So by considering all the factors, the conclusion was that the mutation caused by genome editing, if the mutation caused by genome editing cannot be distinguished from the mutation that happened in nature or with mutation breeding, there is no need to regulate such gene-edited product as GMO. So this is the same summary that I showed you earlier. Now you know rationale behind the GM and non-GMO determination, but because genome editing is a new technology and still evolving, the Japanese government has decided to take a prudent approach and still require developers to voluntarily submit a pre-consultation document, even if the product is not a subject of GMO. This allows the government to accumulate the knowledge and case example, and also to grasp information of commercialization so that they would be able to take actions in case they need one. So here is the list of information which should be included into the notification document to MHLW. Crop variety usage of the product and purpose of the usage, and method of genome editing and description of the modification, absence of foreign DNA, and confirmation that the change in DNA doesn't produce new allergen nor increased non-toxin, and change of the nutrient related to the target metabolic pathway if applicable, and years and month of launch. Information 1 to 3 are mandatory. The information to MOE and MFF is basically the same except for item H. So discussion on possible influence on biodiversity is required instead of the discussion on allergenicity and toxicity. The pre-marketing voluntary notification is a two-step process as shown in this slide. The first, the developers submit a pre-notification document to each competent ministry. The regulator may even consult with their expert if needed, and when they agree that the product is not GMO so that no safety concern, the developers submit the notification document and the document is immediately posted on the website. So this process is very transparent. Next, I'd like to briefly touch on the food labeling policy. Consumer Affairs agencies considered 4-point listed here: consumers' intention, feasibility of labeling, detectability, and international harmonization. Before came up with the policy, the agency held many hearing meetings and invited comment from key stakeholders. Their findings were there are mixed opinion from consumers. There are, of course, many voices requesting mandatory genome editing labeling for consumers' right to choose, but also there are voices not requesting the labeling if the product is not distinguishable from the product through regular breeding because it's confusing and it would add cost. There was a consensus opinion that it is difficult to develop detection method so that the government won't be able to check the truthfulness of the labeling. So consequently, agency determined that the non-mandatory labeling is required for the genome-edited product, which is determined as non-GMO, but voluntary genome editing and non-genome editing label is still allowed. Actually, the agency is encouraging the developer to put the voluntary genome editing labeling, especially for the product with consumer benefits, so that the consumer can develop good image to genome editing. To date, 3 products have completed the notification process, and the first one is Hijab Tomato. The tomato contains 4 to 5 times GABA, so eating just 1 to 2 tomatoes per day helps lowering the blood pressure. So for smooth market introduction of this first genome-edited product, the Sanatex seed company, which is a university-launched venture company, distributed the seedlings to about 4,000 home gardeners this spring. Actually, I also received the seedling and grew them on the balcony of my apartment, and I really enjoyed the harvest. The company has started online sales of fresh tomato and will be selling the tomato puree in December. To ensure consumers' informed choice, the company is putting a sticker on the tomato product shown here and being transparent that the tomato is developed through genome editing. The second and third product are gene-edited fish to support aquaculture industry. One on your left is sea bream with more meat and feed efficiency, and the one on your right is a tiger fish with fast growth and more feed efficiency. The regional fish company, which is also a university-launched venture company, named the fish like 22nd century sea bream and 22nd century tiger pufferfish with a hope that genome-edited fish will be common towards the next century. The company has no plan widely commercialize the fish product, at least for a while, but they solicited internet crowdfunding and will send the fish product to the investors in return. And according to the company, they have raised more than 3 times the amount of their goal. So as a summary, genome-edited handling policy has been finalized in Japan, and the notification process is well functioning so that the 3 products have completed the notification process. There have been R&D programs supported by the government, but not only for that, the government is also conducting some activities to gain the public acceptance of the technology. Positive movement continues, and field testing of low steroidal glycoalkaloid potato and wheat with tolerance to pre-harvest germination is in progress, so that I am hopeful that more product will be notified in the near future. With that, thank you very much for your attention. Thank you.
Thank you, Dr. Miko, for a very, timely presentation. You covered all the areas excellently. Without much further ado, it is a pleasure to introduce my next expert and speaker, Dr. S.R. Rao, who is affiliated with the Indian Agriculture Research Institute from New Delhi and is a visiting scientist in Pretoria University, Japan, and White Experimental Station, Adelaide, in Australia. He served in the Department of Biotech, Ministry of Science and Technology, Government of India, as Senior Advisor to the Government. Currently, he is Vice President of Sri Balaji Vidyapith Healthcare University in Pondicherry, promoting cutting-edge research, innovation, and interdisciplinary development. During the service period in the Government of India for 30 years, he had gained knowledge and experience in dealing with R&D promotion in agriculture, medical, and animal biotechnology, as well as planning, budget, and technical coordination. Other areas include biotech regulation, establishment of sophisticated biotech facilities, promoting international collaboration with developing and developed countries, pursuing public-private partnerships, development of intellectual property rights, and also with biodiversity and biosecurity issues, negotiations in international conventions such as the Convention on Biodiversity and the Biological Weapons Convention. He specializes in core and cross-sectoral policy issues of biotech development, regulation, biosafety, and biosecurity. He has also contributed and drafted and published government guidelines for safety and secure containment, handling of high-risk pathogens, and certification. He's also authored other guidelines as well along similar lines. He's also the co-founder and convener of the Global Alliance for Pandemic Preparedness and Response. He's published 55 research papers in national and international journals and 2 books, each on animal vaccines and diagnostics, and other on biotechnology in Asian countries. He's visited more than 50 countries on scientific and official matters and made more than 100 scientific and technical presentation in cutting edge area. It's really glad to have you here. Dr. S.R. Rao, you have the floor. Thank you.
Thanks for that kind introduction. Am I audible? Hello?
Yes, you are. Yes, you are. Yes.
Yeah, I think it has all been said already. I will not tell about myself. I think my previous speaker from Japan, Dr. Miko, has made my job very simple. The pathway evolving in India on regulatory landscape of genome-edited crops is more or less in the same boat, except for some variations. And next slide. Sorry. So my topics are, I'll give us a little bit preamble, legislations and definitions, review and analysis of assessment of genome-edited crops, approval levels and data requirements, crops under development, and current regulatory scenario. Actually, everybody will agree that regulatory policy, this is one of my favorite things I start with always. Legislations and provisions, scientific complexity and risk assessment. In fact, when genome editing challenge came to the government, we went on the second one, scientific complexity and risk assessment, rather than just sticking to the old legislations, which have got several sentences regulating every damn thing that involves gene modification. Policy of the country, of course, social and ethical considerations, intellectual property rights. I just want to recap people who are aware and who are not aware, just to inform them. Legislational rules are red box. They have to be always considered by the parliaments and assemblies, politicians. It's very difficult to change. Regulations and guidelines can always be tweaked with depending upon the changes in technology. So what we harped on genome editing is basically we tweaked with the level of approvals, decentralized approval system, and also the level of risk assessment and data requirement that are needed. The rest of the process is more or less same as we see in any GMO regulatory framework. So we have also similar problem that environmental protection law, which was drafted in 1989, includes more or less everything, deletion, removal of parts of heritable material, besides new combinations of genetic material. If not new combinations, it includes all other editing, deleting, and also insertion, as has been already been in the definition. So the whole law is preventing us to do any more changes in the law because it's very tough to get act approved by the parliament. Then the Seed Act actually is after the biosafety approvals. Seed Act requires anyway 2 years of performance trials before it is commercially released. For the processing of food and import of food, we have a Food Safety and Standards Act. In fact, I was the chairman for 6 years, and I'm happy to say that the first draft regulations of the technology for import and also processed food has been published 2 days ago. The genome editing crops will also follow more or less same pathway, except there may be some discussion on the labeling part. So this is how the hierarchy is. First, you have a biosafety approval. Then you have a registration of the food. If it is domestic food, if it is imported food, you have some other regulatory pathways. So in general, genome editing is regulated in India as a process and a product, but the level of risk assessment varies. So what the government does typically, it has constituted a committee at interministerial level with experts for policy at national level, and it has constituted subcommittees for genome editing of plants in green, and then humans and animals, and also committee on biosecurity. Incidentally, there is no controversy on human and animals because everything is regulated there. In case of genome editing of plants, there are some debates going on still, even though regulatory framework and the guidelines are made very clear. This has been really very well explained by Dr. Miko before me. I think that saves my time. The SDN, SD1, 2, SDN3 concept holds true for India also in terms of regulatory approach. Then the committee had a challenge whether the full regulatory oversight is needed or exempt SDN 1 or 2 or SDN 1 or 2 completely. So there was a lot of dialogue going on among these options, whether to exempt some regulations or include all of them. So then they drew a very nice diagram that there is as far as SDN 2 is concerned, it falls in the wall. It falls on the wall between GMO and non-GMO type of approach, whereas SDN 1 typically has a point mutations and deletions, which falls under the regular mutagenesis and natural mutations. So therefore, they said that can we deregulate SDN 1, yet keeping respect with the law, or regulate, of course, SDN 3, like in case of genome editing. Depending upon the complexity at genotype, phenotype, and biochemical level, we will have a data requirements. This is what this group of committees decided. They also draw this diagram. The risk scenario will increase as we go from SDN1 to SDN3. I will not go into many details, which Miko has explained already. So we have also 3 levels of committees. One is Institutional Biosafety Committee. All activities are regulated at institutional level. Review Committee on Genetic Manipulation, which does the risk assessment and recommends approvals or no approvals to the higher committee. These 2 committees are basically controlled by Department of Biotechnology, Ministry of Science and Technology. Then Genetic Engineering Approval Committee, it thinks about a large-scale environmental release. I happen to be the member and of these committees for a decade. So Ministry of Environment and Forest deals with this particular Genetic Engineering Committee. Then it goes to the Agriculture Ministry for declaring for the seed registration. So these are the levels to commercialization, starting from the institute to the Genetic Engineering Approval Committee for biosafety releases, then followed by Agriculture Ministry. So these are the data requirements in summary, which the group of experts decided, along with our internal experts, that when you want SDN2, SDN1, even though it is a green box, you need some basic requirements about the, as been showed by Miko, that some important information is required for notification at the institution level. SDN2, of course, we also moderated that SDN2 case by case, the risk assessment data requirements will reduce or increase. SDN3, of course, are treated like GM organisms. But overall, there have been lots of articles disturbing the committee of experts, and some saying mutations, some saying unintended mutations, even in SDN1 related changes. So therefore, the document also says possible off-target effects, some sort of an evidence through database or bioinformatics has to be given at these levels of transcriptional, post-transcriptional level, as to there was no unintended mutations. Then, meanwhile, there was this document was basically worked out by me for 6 years. It was released at the BWC Convention of Geneva because it has got a lot of risk assessment procedures. This is a very simple to explain. Sorry. In case of genome editing, the institutional biosafety committees can permit all operations of SDN1 type. That is a decentralization process of regulatory framework. And then authorization for SDN2 and SDN3 for further consideration by the upper apex bodies at the national level. So now I'll go to the crops. These crops, Government of India has got an agreement with US laboratories for training our people batch by batch. Every year, almost about 50 people have been, faculty and students have been trained in genome editing technologies across organisms. We also have a definite funding every year to do R&D and develop, keep ready the products while we are evaluating the regulation by consensus at the government and also at the public level. So I just give you what are the centers and what are the traits that they are attempting. There's a huge list of it. Groundnut, for example, aflatoxin resistant, the usual suspects of these traits in a developing country or in a agriculture country. Chickpea, for example, dry root rot, seed size, pearl millet, rancidity. Then we have another institute called National Institute of Plant Genome Research. They are in advanced stage about, they developed a low glucosinate mustard. It is an alkaloid. Its level has to be controlled as for the food safety. And it results into more edible feed also for the cattle. Again, chickpea is the same trait. Maize is also attempted at this place. So if you say advanced stage is only this particular mustard crop, then tomato, of course, the Bose Institute in Calcutta, they are working on regulatory gene expression basically for use in synthetic biology approach. Then there is an agriculture university which works on oleic acid and low linoleic acid. Rice, again, nitrogen use efficiency, water use efficiency at the place where I had my PhD. And International Center for Genetic Engineering working on rice for low phytate nutrient use efficiency. And National Research for Plant Molecular Biology is working on mustard again. Tamil Nadu Agriculture University is working on disease resistance. National Agri-Food Biotechnologies. This is a very interesting one, banana. This is a B carotene rich banana they are developing. Earlier they had technology imported from Australia. Now they are going for genome editing. One interesting part of the government expert committee decided was, don't call genome edited crops as GMOs. You designate them as G-ed crops, G capital, E capital, D small, so as to distinguish these two categories of intervention of genes. Then Institute of Life Sciences in Orissa state, this is also working on a traditional banana, which has got lots of seeds. So they are halfway through successful. Many progenies are available now, which are seedless. It is one of the most preferred banana for nutritional purposes. The National Botanical Research Institute has tomato, cotton, rice, and chickpea. Now, Mike was kind enough to invite me. I thank the ISA all for opportunity. He also told me, can we put it in this simple diagram like this? But I had some problems in putting it. I took the Australian pattern of regulation and see where I can add some of these evolutionary guidelines that are going on in the country. You can see in case of SDN1, you require still a substantial equivalence and phenotype composition data. But what saves is, it will be all done at the institution level. There is no need to go to the hierarchy. Then in case of SDN2, also limited regulation as a GMO, but case by case. So you require all these substantial equivalence data, plus some field trials. Here institution field trials in there are enough. And whereas in SDN3, you have to go through all the procedure as that of GMOs. But for that, the approach is more or less similar to Japan and Australia. So finally, all these things, animals and humans, have been already taken for implementation. In fact, I am chairman of a committee for genome editing of gene therapy. We are evolving separate guidelines for the clinical trials. Now, as far as crops are concerned, these are the suggestions. The controversy is, many activists, when it was kept in public domain, have been raising that SDN 1, 2, 3, irrespective of anything, you have to regulate in the same way as that of GMO, and which is expected from them. And the government is also a bit panicked to see how to go about. So which path government of the day takes, apart from what the typical decentralized regulation and complexity-based risk assessment suggested by the expert committee, to regulate all or regulate based on complexity of genome editing. Thanks to all ISA and also Dr. Michael Jones for especially inviting me. This is from the, I think this is one of the sequencing centers in US. I like this because which path we take, we don't know. That is the status in case of genome editing in India. Thanks for the opportunity.
Thank you, Dr. Rao, for that presentation. I'm grateful to all speakers that they've been sticking to their time limits, and thank you for your very clear illustration of the regulatory landscape. Without much further ado, it is yet again my pleasure to introduce the next speaker, Dr. Shahid Mansoor, who is the Director of National Institute of Biotech and Genetic Engineering in Pakistan, Faisalabad. He is a Sitara Imtiaz, which is a national honor. He's the Higher Education Commission Distinguished National Professor, as well as a Fellow of the Pakistan Academy of Sciences. He has been leading the charge of the Department of Virology at NIBG. Other than that, he is actively consulted by the government, the public, and the private stakeholders related to biotech, commercialization, as well as regulatory affairs. Dr. Shahid, it's a pleasure to have you, and you have the floor. Thank you.
Dr. Mansoor, please unmute.
Okay, sure. Unmute okay?
Yes, it's
Yes,
okay.
yes, we can hear you.
Okay, you can hear me. So thank you, Dr. Adil, and ISA for inviting me to give a perspective about Pakistan. So I will mostly talk about the research because, my earlier colleague from Japan, Dr. Miko and Dr. Rao from India have given very elaborate mechanisms. So I think that, probably it is the similar situation in Pakistan, but I'll give you some perspective about the policy. But mostly I'll focus on research because, we have an emergency in Pakistan of food security. And also there's a lot of capacity in the country because of the work that we were previously doing on genetic engineering. So obviously, the protocol for transformation for developing gene construct was available. So I think we just use that ability to transfer the technology and acquire the genome editing technology at NIPD. So this is a slide about the situation in the country that we need around 37% of population is food insecure. Then there's the problem of stunting, wasting, underweight, and also there's a huge problem of malnutrition, which is resulting from the deficiency of micronutrients such as vitamins and minerals. So obviously, the genome editing provides new tools to come up with some of those issues. So what we did is that we made a kind of a national consensus by inviting all stakeholder and then institutional, and by discussing with all colleagues, we have identified some of the crops and the traits that are important for the country, like for rice, obviously yield, better grain, nutritional value, herbicide tolerance. As I mentioned earlier, that nutritional value is very important for the country. So this was one of my important area that we have focused. In potato, we have previously done a lot of work on engineering resistance by genetic engineering. So now we have switched to using genome editing technologies, which is more acceptable to the regulators and probably to our national, general public. So in potato, we are working on virus resistance, sweetening control, and I'm glad that we have some of the very good collaboration with Journalist Center with Professor Jones, where one of my students is working for sweetening control and virus resistance. Then wheat is a very important crop, is a political crop in our region because it's something related with the food security. In northern India and Pakistan and Afghanistan, wheat is our major. So if you have shortage of wheat, then it's a big problem. So we are working on definitely yield because with the elucidation or availability of genome of wheat, which is a hexaploid, then there's a lot of genes have been identified earlier. I will give you some of the example how the information from rice genome has been translated into wheat. So here we are working on disease resistance and then on yield increase. So I'll give you some of the nutritional value. So again, you can see that everywhere we are emphasizing on nutritional value. In case of cotton, disease resistance is one of the important area. Then better quality nutritional value, oilseed crop, higher yield, better quality nutritional value. Again, here we are also working on some of the anti-nutrients. So by editing some of the genes which are cause of nutritional value, like erucic acid and glucosinate. So we are funded by ICGEB with a project to work on this improvement by silencing or editing genes associated with ethnic nutrition. Sorry. Okay. So I will not go into the details because most of the audience is already aware of the system which are available. But I must emphasize here that there is a lot of progress in our tool development. So in the next slide, I'll show that there are different versions of Cas protein, and so we have adopted different tools like Cas9 and then Cas12a for editing. Recently, we have also there's a great interest in a compact form of genome editing, which is done by Cas12f1. And this we have recently written about this in Trends in Plant Sciences. So obviously, our area is that to enhance yield, improve quality, disease resistance, herbicide tolerance, and one of the some of the other tool like Cas14a and Cas13, we think that these are very wonderful tools for editing virus resistance, especially Cas14a, because it targets single-stranded DNA. So I think that and then some of the Cas which target RNA. So this could be some very important tools in our ability to edit crops. So I'll just give few examples what we are doing before we go into the regulatory affair in Pakistan. In cotton, as you know that cotton is our mainstay of economy. We have been using, cotton is the only crop, genetically engineered crop, which is allowed in Pakistan. And recently we have developed our own new events for insect resistance and herbicide tolerance. But there are many challenges, and especially in climate change scenario, growing cotton under very hot condition is a challenge. So we have identified different targets for drought resistance, salinity resistance, temperature tolerance, and heavy metal resistant radiations. And we think that by having a combination of genes for abiotic resistance, disease, and also biotic resistance, we can come up with good quality and much more environment friendly growing of cotton in the country. So there's already a lot of information available about how much impact the GM cotton has created in terms of environmental protection. So this is not related to the topic, but I think that there's another very interesting case where rice is both a staple crop food in Pakistan, but also an export commodity from Pakistan. So we have identified some of the genes which are negative regulators of some of the important traits. So these are like so rice yield is driven by number of panicles, number of grains per panicle, and grain weight. So we have identified 4 different genes which are negative regulator of tillers. But I must say that these are beside identifying these negative regulators, we are also working on the positive regulators. So beside genome editing, we can also use genomic selection to identify markers. So by having a combination of both genomic selection and genome editing, we hope that to enhance the production of rice or improve the livelihood of our farmers. The second case is again we have identified the homologue of these genes in wheat, which are related to grain quality, grain yield, and then the tiller number, and all this approach, as I said, we are just making a copy of that approach that we are following in rice. Is that we are beside genome editing, we are also using other technologies and other approaches to have genetic gain in wheat. So these are some of the example, how we have done this. There are challenges, especially in case of wheat, because it's a hexaploid plant. So obviously, we have to confirm that all alleles are modified or edited in case of wheat. There's some work on disease distance as well. So I must say that genome editing has provided for the first time, direct access to, modify or edit those genes which are related with yield and quality. So there is also some work on disease resistance. For example, we have already done this work, and this work is under preparation for publication. So here what we have done is that, we have targeted one of the elongation factor for virus resistance, and we have challenged this plant. This is a potato, genome-edited potato. And as I said earlier, that beside this, so these plants are now in our tunnel, and we have tested them against the PBY, and we hope that we can also check whether this also give resistance against other viruses or RNA viruses in case of potato. This is something which is already published. This is a kind of a taking the work that we did earlier to identify some of the susceptibility genes. So we published one paper in Trends in Plant Sciences about the susceptibility genes, and in rice we have identified some of those sweet genes, sweet 13 and sweet 14. Here we have shown work where we have edited sweet 14, and these plants have been tested against for resistance, and we found that these plants are resistant. So that gives you another tool beside breeding for XA genes. So there's now we have a choice of whether to use XA genes which give resistance against bacterial bite or use the genome editing. So there's also a nice work done. So we hope that by targeting both sweet 14 and 13, it can result in broad spectrum. So again, in the regulatory affairs, we have to see that how it is taken up by the authorities. Also, what we are trying to do is that also use, especially in case of wheat, speed weeding approach to speed up our genetic gain work in this. So this is about one slide about regulatory affair. As I said earlier, that the presentation from Dr. Miko and Dr. Rao has pointed out about the regulatory affairs. So there's a very positive attitude. Adil can second this, that we are very positive about the technology, and there's a very enthusiastic response from research community and funding agencies. So Ministry of Science and Technology, Ministry of National Food Security, and Higher Education Commission, they are all funding research project. And there's a because we have a huge number of scientists who have been trained abroad, young scientists, and they are coming back and and starting this work. So in the country, there are several groups that are working. So as I said, that because the capacity was already existing in the country, because there's a lot of investment made in training scientists and having infrastructure. Almost all research institutes and universities have a biotechnology department, and there's someone who is interested in genome editing. So for the regulatory affair, we are very clear that we are very much thankful to the policy which is in Japan and also in Australia, and I think that Mike Jones will talk about this. and that we are very much supportive of the very practical approach which has been taken up by these countries. And I think that we'll probably share with these two countries about and have a brainstorming session with these, so that we apply the same thing here. So at NIBD, we invited all the expert and technical advisory committee of the National Biosafety Committee. I co-chair technical advisory committee of NBC, so obviously there's a very positive response. And so just like Japan and India and other countries, we decided to have a case-to-case basis approach for approval. And also there's an emphasis on tissue culture independent approaches, like for example, so that we don't have to transform. So recently we have been working very closely to use viral vector for delivery of smaller versions of Cas protein, and also use viral vector for delivery of guide RNA. I think, and also using the nanotechnology, so that the time is reduced, so we don't have to go back and backcross and remove Cas protein. So I think that this approach probably is and there are a good number of research papers available now to using viral vectors. And I hope that by having all the scientific community on board and funding agencies, I hope that there will be a lot of good news coming from Pakistan regarding adoption of this genome editing technology. With these words, I must thank all the organizers, and this just take that we need this technology. We need actually the only way that we can progress is to have the technology available, and also use the technology in conjunction with other tools which are available. And there's a positive and enthusiastic approach exists in Pakistan. Thank you very much, Dr. Adil and all the audience. Thank you.
Thank you, Dr. Shahid, and to all the presenters for this sessions. Thank you for sticking to the time and really, hitting home the point on what the regulatory landscapes are in your respective countries. So we have around 7 to 8 minutes for the question and answer session. So what I'll do is and I'm also grateful to Dr. S.R. Rao for already answering some of these. So in the chat, we've got some questions that I'll put forward to my panelists. First is a question that is directed at Dr. Bizon. In China, is there any regulatory policy at the local government level? Dr. Bizon, would you respond to that? You're on mute.
This me?
Yes, here we can hear you.
Okay. So what's the question?
Yeah, so it says that in China, is there any regulatory policy in local government level?
No.
Okay.
All the policy is issued by the state council or the Ministry of Agriculture.
Thank you for the prompt response. And in many cases, it's also dependent on the federal or the provincial legislation, whether it's federal subject or not. We have a lot of questions related to like Japanese regulatory landscape. Dr. Miko, if you could answer. So one of the questions is that should the Japanese government promote public acceptance of private commercial GM products using public resources?
Maybe I didn't explain very well. The Japanese government is not supporting any commercial activities for the private company. What the government does is to support the research, so that for the Hijab Tomato case, they did a proof of concept type research. And after that, it's a private company's responsibility for product development and commercial activities. Actually, what the government is promoting is educating the Japanese consumer what the technology is, what the technology looks like, as well as why what sort of the application that genome editing technology may help to keep advancing the Japanese agriculture under the current global warming substance and all sort of things, so that they are not supporting the private companies' commercial activity is what I would like to say.
Yeah, thank you so much for your prompt response. I've got a very interesting question. So it says that, and it's directed at any of the panelists, regarding the regulation of gene-edited crops. If each country has their own system, is there any common universal regulation that should be followed? Is there any copyrights when using a gene editing tool like CRISPR system if adapted in any lab in any country? So I think harmonization is a goal that all of us desire. So if any of the panelists can, take the lead on this, that how we can, actually proceed towards harmonization.
Okay, Dr.
Yeah,
Rao.
Dr. Rao, please go ahead.
Actually, it's the whole regulatory landscape of genome editing crops, whether to call gene editing or genome editing, is also, everything is evolving. So each country has got already fixed notions, and some of them are evolving. So the crops are also evolving. We don't have a great experience with the new technology. But globally, there is a view to harmonize at least at the risk assessment level, because you can't change the legislations of the governments, because you can't harmonize Japanese law with our law, right? You can always do risk assessment harmonization. The data requirements can be harmonized. I think with the GMOs, we have that experience, and Cartagena Protocol or the food, what is called Codex Alimentarius for food, and also the OECD, most of them have brought some sort of a harmony in the data requirements, and genome editing are no different. We are depending on the same data requirements. The only difference is that what is the level of data requirements depending upon the complexity of the technology need to be addressed by country, and that is up to their own ecosystem of activists, governments, understanding, scientists, capacity, so on and so. So that is what is it. Otherwise, harmonization at risk assessment level is extremely possible, and it's very easy to do. And secondly, in India, we also give funds to small private sector for who's called Small Business Innovative Research Initiative to develop genome edited crops. Unlike Japan, we have that mechanism also to transfer the technology. We also do the other way. And that's what. What is the second question?
So the second question was, is there any copyrights when using a gene editing tool like CRISPR system if adapted in any lab in any country?
Of course. Of course. One has to have a freedom to operate whenever you touch these technologies. Even the kit that you have, it is already clearly written. And the PCR, of course, now, it was also written in the PCR kit earlier. Now it's slightly away from the patent regime, what is the older systems. So all these techniques are having patent implications. But one caution is, one advantage is there. Patent is a property right of the country. Unless that particular patent is also registered in India. For example, many transformation technologies of rice are registered in several countries. So you have a transformation itself, or tissue culture itself sometimes is patented as a process. So if it is patented in your country, be cautious about using the technology. As far as it is not patented, inshallah, you can enjoy the technology. Yeah.
Thank you, Dr. Esarov, for that very elaborate response. And like harmonization, it's a nexus of science, policy, and regulation. Dr. Shahid, we have got a question directed at, does Pakistan work on crop nutritional value, including biofortification? What aspects of nutritional value are targeted? I know the answer is yes, but can you further elaborate on what nutritional value that we are targeting?
Yeah, I think that, this is something that, we have in recent year, we have emphasized that our crops are deficient in some of the essential nutrients like, zinc, iron, and then vitamins. So these are the core, area where we think that, zinc. So there's already emphasis on zinc and iron fortification. And so we are working some of the Australian counterparts, and also within the country that we should work on. So these are The other target is, which is a little bit more complex, is vitamin A. That's another area that, instead of having, say, golden rice, which is genetically modified, as I said earlier, that Pakistan is a major exporter of rice. So obviously we have problem adopting GM rice, but this is something, biofortification about vitamin A, which is possible. India has also given an example of banana. I think these are all very interesting. One thing that probably ISA can do is that, at least these developing countries can share, or at least for testing and trials, for example, the healthy tomato from Japan or healthy banana from India. So those are the things that all developing countries have a common problem of malnutrition. So obviously, we have to have some kind of working together, and probably ISA can play a role. And then we have Adil and Professor Mike Jones, who are kind of they know everybody in this area, and I hope that we'll come up with some of at least some commonalities and work on those. Dr. Rao has already very clearly identified about the patenting. So the same apply for Pakistan. Thank you.
Yep. Thank you. So without much further ado, I'll hand over towards the next session to Dr. Rodora. But before that, I'd like to thank all of the panelists for their wonderful presentations. They were very illustrative and very targeted. And I would also request you that the questions that we could not take due to the paucity of time, if you could, answer them directly in the chat. It will be really good help for the audiences. With this, I hand it over to Dr. Rodora. It was an absolute pleasure. Thank you.
Hi, good morning, good afternoon, good evening, everyone. This is Dr. Rodora Romero Aldemida. You can call me Ola. Thank you very much, Adil, for this wonderful session. And we also, I would like to again reiterate his request that our speakers, our panelists, please respond to the questions in the Q&A box. You can write directly to the Q&A box and also at the chat. Well, this session, the next session is going to be on the industry perspective. And this is, again, the title is still Research, Policy Approaches and Implications for Market Trade. And this is the industry perspective. So we'd like to start with a friend. He is Mr. Osman Newett. He's the Chief Executive Officer of the Australian Seed Federation, the peak industry body for the Australian seed industry at the local, state, national, and international level. He was formerly the CropLife Australia's lead in policy, advocacy, and regulatory reform activities relevant to the crop biotech sector in Australia. He received his bachelor's degree in science, in laws, and a graduate of the Australian Institute of Company Directors. Please welcome Mr. Osman Newett to give us the presentation on modernizing and future-proofing the Australian gene technology scheme, a seed industry perspective. Welcome, Dr. Osman.
Thank you, Ola. Thank you very much for that kind introduction. And thank you very much to the organizers of this webinar for your invitation for me to participate today. So I will just share my screen. So as Ola said, my presentation today is around modernising and future-proofing Australia's gene technology regulatory scheme. I've split my presentation up into three separate parts. To begin with, I'm going to tell you a little bit around some of our reviews of our gene technology regulations and our Gene Technology Act, and just so you can understand where we currently sit in terms of our regulation of gene editing. I'll then take a little sideways dive into food derived from new breeding techniques, because there's some interesting regulatory reviews happening in that area at the moment. And then I will come back to the Gene Technology Act and the Gene Technology regulations and give you my opinions and my suggestions from the Australian seed industry around how we could do things better and how we could better modernise and future-proof our gene technology regulatory scheme. So we're going to look back into the past first. actually. First, I'm going to tell you about a little bit about how we regulate gene technology in Australia. And so the import and cultivation of GMOs in Australia is regulated through a nationally consistent scheme. At the centre of the scheme is, I'm just going to pull up my laser pointer, is the gene technology regulator. And the gene technology regulator is responsible for administering the Gene Technology Act. Underneath the Gene Technology Act sits the Gene Technology Regulations, which I'll be talking about in my next couple of slides. So the Gene Technology Regulator is responsible for administering the Act and is also responsible for making decisions on whether to approve field trials and also whether to approve the commercial release of GM crops. GM products fall under a whole bundle of different regulatory agencies. Of particular interest to this presentation is Food Standards Australia and New Zealand, which is responsible for setting the standards for the safety, content, and labelling of food in Australia, including foods derived from gene technology. So I'm now going to ask you to cast your mind back to the dark old days of BC, or before COVID. This was 2016, and the Gene Technology Regulator commenced a technical review of the gene technology regulations. This is what we understood before this review took place. Natural mutations and mutagenesis were not gene technology, but inserting transgenes or transgenesis was gene technology and resulted in the creation of a GMO under Australia's regulatory scheme. We then had these new technologies coming onto the market. They were quite often called new breeding techniques or plant breeding innovations. What we understood about some of these techniques was that some of them were closer to those mechanisms that were not gene technology. Some of them were closer to transgenesis, and some of them were somewhere in the middle here. And we weren't sure whether they fell into the green box or the red box. What we did understand about these new technologies was the feature of SDN1 was that it relied on non-homologous end joining, whereas SDN2, ODM, and SDN3 relied on homology-directed repair. The regulator decided early on that SDN3 would be considered transgenesis, and so that was not part of this review. However, the review focused on which box do we put these technologies into? SDN1, which relied on non-homologous end joining, and SDN2 and ODM, which rely on homology-directed repair. So at the start of the review, the status of these technologies was ambiguous. Some were excluded, some were uncertain, some were regulated as GMOs. At the conclusion of the review, we had some clarity over the regulatory status of these techniques. The regulator decided that SDN1 would fall into the green box, and organisms created using this technology would be excluded from regulation in Australia. However, the regulator decided that SDN2 and ODM would fall into the red box and be regulated like SDN3 and transgenes as GMOs in Australia. So just to summarise, organisms modified using site-directed nucleases without templates to guide genome repair are not regulated as GMOs in Australia. These organisms are treated the same as those that result from the conventional breeding process. And importantly, and different to some of the other presentations we've heard today, no consultation with the regulator is required. However, if you use a template to guide genome repair, for example, SDN2 or SDN3, then the resulting organisms are GMOs, as are organisms modified using oligo-directed mutagenesis. So that was the gene technology regulations. In late 2020, the Australian government launched a consultation paper on modernising and future-proofing the National Gene Technology Scheme. So whereas that initial review I've been speaking about was a technical review about the regulations, this review was a policy review of the Gene Technology Act. And so I guess you could say it was at a higher and broader reaching level than that very specific technical review. This December 2020 paper presented 3 options for regulatory reform. Option A was to keep the status quo and make no changes. Option B was a risk tiering model, where dealings with gene technology would be classified according to their indicative risk. And option C was a matrix model, where the nature of the dealing determined its classification. For my presentation today, I'm just going to focus on option B, the risk tiering model, as this was the option that was preferred by the agricultural industry submissions to this review, and it ended up being the option that was preferred by the regulator and the government when they came out with their recent decision paper. So let's take a bit of a deeper dive into risk tiering. So this option B enabled dealings with GMOs to be distinguished on the basis of indicative risk. Now, I like this because this enables a proportionate risk response, and regulation should always be commensurate with risk. It enabled dealings with GMOs to be placed into 3 separate categories: non-notifiable, notifiable, or licensed. And then even within this license category, depending on the indicative risk, the product might be subject to either a full assessment, an expedited assessment, or a permit. So under this model, the gene technology used to create the GMO would be a relevant consideration. So if you had used a specific type of gene technology, for example, a specific type of gene editing, and this was found to present a very low risk and a case-by-case assessment is not required, then this dealing could fall into one of the lighter touch pathways, for example, the non-notifiable or the notifiable pathway. However, I still had a problem with this approach because under this option, even those dealings that are classified as non-notifiable are still considered a GMO. They're not excluded from regulation. And this is significant compared to the SDN1 exclusion that I told you about earlier. So this is all about products that you're intending to release into the environment for field trials or commercial release. But what about food derived from new breeding techniques? So the Gene Technology Act and the Gene Technology Regulations regulate live and viable GMOs intended for release into the environment. But in Australia, we have a different regulatory agency for GM food, if you can remember back to my opening slide that had the diagram of our regulatory system. So GM food is regulated by Food Standards Australia and New Zealand, and they've recently commenced a review on how the Australia and New Zealand Food Standards Code applies to food derived using new breeding techniques. So what this means is that for practical purposes, in Australia, a product developed using SDN1 is not a GMO for cultivation. However, if it is a GM food, would be subject to the outcomes of this FSANZ review. And this is what the current definitions in the food standards codes look like. If you've bred a new food using either crossbreeding, cellular tissue culture, or classical mutagenesis, as long as you've met the overarching requirement that all food is required to be safe and suitable, then you've got a clear path to market for your product. However, if you've used gene technology to create your food, then your product is captured for pre-market safety assessment and delayed from entering the market. So what FSANZ have put forward for consultation in their current documents is that the need for pre-market assessment of food derived from MBTs is essentially a question about risk and how MBT food compares to conventional food. So FSANZ have said that if it can be demonstrated that MBT food is equivalent in risk to conventional food, then you can argue, and I agree, that a pre-market safety assessment is unnecessary because you have to ask yourself the question, what are you regulating and why are you regulating it? FSANZ have also said that when assessing the risk from MBT food, all of the size of the genetic change, whether it was intended or not, or the method used to affect the genetic change are all irrelevant considerations from a food safety perspective. Again, I agree with these statements from FSANZ. So FSANZ are saying that the crucial factor from a food safety perspective is the impact of that change on the food. So if you've used a new breeding technique such as gene editing to create a genetic change, and it hasn't resulted in a new or altered product characteristic compared to conventional food, then FSANZ concludes, and again, I agree, that the MBT food will carry the same risk as the equivalent conventional food. And this provides a clear basis for excluding these foods from a requirement for pre-market safety assessment as a GM food. So if you cast your mind back to that diagram I put up earlier, these foods that carry the same risk as the equivalent conventional food, they should have a clear path to market. They shouldn't get caught in that net of pre-market safety assessment when there is no justifiable food safety reason. So the final part of my presentation, I'm coming back to the Gene Technology Act and the Gene Technology Regulation, and I'm giving you some of my views on how I think the regulatory approach in Australia could be improved and better modernised and more future-proof. So the International Seed Federation, of which the ASF is a member, has created the following policy principles for the scope of regulatory oversight for plant breeding innovation. And they have said that plant varieties should not be covered under the scope of existing biotechnology regulations if one of the below criteria is met. So if there is no novel combination of genetic material, for example, there's no stable insertion in the plant genome of one or more genes that are part of a designed genetic construct, or the final plant product solely contains a stable insertion of genetic material from sexually compatible plant species, or the genetic variation is the result of a spontaneous or induced mutagenesis, then these products should be exempt from the scope of existing biotechnology regulations. And so what we've seen is that the range of tools and applications of genome editing are continuing to grow. 5 years ago, when the OGTR commenced their review of the regulations, we were focused on SDN1, SDN2, ODM, and SDN3. You could almost call those technologies old technologies now, as the spectrum of these applications has just skyrocketed in the last few years. And so what this says to me is that there's a need for regulatory approaches to remain flexible, to allow for advancement in technology and methodology. And this will allow breeders to use these innovations more effectively to make use of existing genetic variation in plant species. So these ISF criteria for the scope of regulatory oversight provide that needed flexibility to define when the resulting products should not be in the scope of existing GMO regulations. Coming back to risk tiering, and does it go far enough? So this comes back to the concept of risk proportionate regulation, and that regulation should always be commensurate with risk. So it's my opinion that the exclusions in the gene technology regulations need to be more outcome focused. At the moment, they're too technology specific. And independent of the technology used, if there has not been any integration of one or more genes in a defined construct, then this should be excluded from regulation. And the reason I say this is that while any form of mutagenesis can introduce risk, including things that we treat as conventional breeding, the use of gene technology for targeted mutagenesis doesn't automatically generate a risk any different to that which arises through spontaneous or induced mutagenesis. Look, it may, but it doesn't automatically do that. So from a risk perspective, it doesn't make any sense to regulate these targeted mutagenic products purely on the breeding process used. So I think there needs to be immediate exit points in Australia's regulatory scheme for products that have been developed using gene technology, but are not a GMO or of such negligible or low risk that regulatory oversight is not required. And so what's happened here in Australia is that by treating products developed using techniques such as SDN2 and ODM as GMOs, we've created international barriers to trade for our product developers. And this is as a direct result of the non-alignment of our regulatory system with those of our trading partners. So our regulatory approach has actually, I guess, put a dampener on innovation in this space in Australia because we do not have alignment with some of our key trading partners. So what do we want as a seed industry? Look, I think that option B, that risk tiering approach, it's a good approach, and it makes sense to streamline the regulation of long established traditional gene technology, or you could call it conventional GMOs, in a way that's more proportionate to the risk profile of well understood and characterized organisms and traits. Now you have to remember, we've been cultivating GMOs in Australia for over 25 years now. We have a quarter of a century of experience with this technology, and we have some very well understood organisms and very well understood traits that it makes no sense to continue to regulate them in the same way that we did 25 years ago. This is why I believe that option B fails to satisfactorily address the different risk indicators presented by gene technology innovations, and particularly those innovations that have a risk profile that we can compare to that of conventional breeding that, doesn't require a pre-market safety assessment. So the ASF advocated for the adoption of an enhanced option B, or a better option B. So in addition to what was proposed, the risk tiering, which is good, but it deals with historical technology, not new technology, we want to specifically and immediately exclude products developed SDN2 and ODM from regulation as GMOs in Australia. And what we also want to see from our regulation is a pathway for the exclusion of new gene technologies in the future. Because I believe that innovations enabled by gene technology, as opposed to GMOs per se, are the future of Australian agriculture. It is really important that Australia has a supportive regulatory environment and that all of these reform efforts that we're all putting so much effort into over several years, it's important they result in a regulatory paradigm that's based on risk indicators that don't automatically treat all products of gene technology as a GMO. Because what we've seen is that this can result in real-world negative outcomes for innovation, trade, and commerce. The risk indicators that our regulators use, they must have a basis in science. And as I said, we have 1/4 century of experience and knowledge and science of the commercialization of some of this technology. It's very, very well understood. So in conclusion, in my view, Australia has gone from a leader, BC, before COVID, to a laggard in 2021 regarding keeping pace with the global trends of gene technology regulation. And we've even seen that today through the presentations you've heard before mine. So we support a risk tiering model. However, it doesn't provide any clarity or pathway forward for the exclusion of gene technologies where the outcome of those technologies is identical to that which could be achieved through conventional breeding. So to truly modernize the regulatory system in Australia, regulators need to consider risk proportionate regulation of new technologies, from those that we know about, your SDN1s, your SDN2s, your ODMs, for more than a decade, to those that haven't been invented yet. And we need to avoid undue regulatory burden where there is no evidential basis for risk to human health and safety or the environment. Thank you, Ola.
Thank you so much, Osman, for this exhaustive and very clarified presentation on what's going on in Australia. I was just thinking that the last time that we heard about the Australian regulations, it is being still revised. So I hope the revision will move forward as what you have suggested. But we have a lot of time for other people to please put in your questions at the Q&A box. I know that these are very interesting presentations. So we'd like to move on. Please, Osman, wait for a while. We will have a discussion after the presentation of Dr. Keiko. Okay, so let's welcome in Dr. Keiko is the BASF Regulatory Affairs Manager responsible for managing all regulatory aspects of biotech events in Asia Pacific. He is a trained entomologist with the Australian Museum and worked in pesticides toxicology and now in biotechnology. Let's welcome Dr. Keiko for Gene Editing Policy in APAC.
Thank you. Thank you so much. And I would like to thank the organizers for giving me this opportunity to talk to this audience. It is a privilege for me. So I will now share my presentation. Okay. Okay, so is that okay? Can you see that all right?
Still not shared, sir? No.
Oh, sorry. I will try again because I saw that.
There, yes, it's
okay?
already on. Thank
Excellent,
you. Yeah.
Thank you. Yeah. So I will be talking about gene editing policy in Asia Pacific and the possible impact on commercialization of gene edited products. And we've already heard a lot of technology being developed in some parts of Asia Pacific, and I'll talk about how policy might impact how we bring some of these products to market. So the global trade of agriculture and food products is pretty varied, with countries importing and exporting to each other, to and from each other, and the amounts they trade between countries, and also the type of products they trade is pretty varied, but generally there's a to and fro exchange of traded items between countries. Yeah, and this picture just shows the various amounts of trades going on between countries. When it comes to trade in biotech products, however, like GM crops, the picture is more lopsided. The main GM products being traded today are soybean, corn, cotton, and canola, and they come generally from the Americas to countries in Asia and the EU. And this trade in GM crops has been going on for years, since the late 1990s. So the figures shown in this slide are from 2016 for soybean trade, but the figures change from year to year, but the picture is generally the same. Yeah, and it is also the same for trade of the other main GM commodity crops of corn, canola, and cotton. So the general pattern is that the GM crops, they go from the Americas, both North and South America, where they are cultivated, to markets in Europe and Asia. In Asia, the main export markets are mainly China, Japan, Korea, although Indonesia and Philippines are now becoming more important GM commodity markets. So when trading GM crops, regulatory approvals are a necessity, and they are in place for all currently traded products in the export countries. Before they commercialize a GM crop, so a company like mine, companies have to analyze the trade patterns and identify key export countries and make sure approvals are in place. Yeah. Otherwise, they face the risk of the products being rejected. So that they analyze the trade patterns, analyze the key export countries, and make sure that they don't face any risk of product rejection. But to get this global approval in the key export countries, to get all the regulatory approvals is an expensive affair. And there was a study some years ago that it costs about $136 million to bring one of our GM events to the market. Yeah, and these figures are from about 11 years ago. So today, probably the cost has gone up. There is a crucial difference between launching and commercializing a GM product compared to a gene-edited product. Because of the high regulatory cost to launch a GM product, you need to commercialize your event in a broad acre commodity crop where the seed market is big enough so that you can recoup your investment. That is the $136 million that I just mentioned. And that is why today the main GM crops are crops grown across huge broad acre hectareages, yeah, like soy, corn, cotton, and canola. However, if there are no high regulatory costs for gene-edited crops, you don't really have to invest in commodity crops. If regulatory costs are minimal, you can afford to invest in minor crops such as vegetable crops. And this is already happening, as shown by the Hijab tomatoes and the sea bream that Dr. Kasai mentioned earlier on. In the US, there are many companies on the verge of offering a great range of GE products. If you go to the USDA MI regulated site, you will see this listed. And on this slide here, I show a screenshot from their website. In the US, there's also a company called Calyx, and they've also commercialized a high oleic soybean, which they developed using talent and not CRISPR. Yeah, a lot of people are working on CRISPR these days, so this product has been developed from talent, which is an older gene editing type of technology. So in this case, although the crop is soybean, they can control the marketing of the crop through strict identity preservation, yeah, because of the high value of the crop. So if you have a run-of-the-mill commodity crop, you can't do that. So in those countries like in US and Japan, where there are certain GE crops, genetic products, which are not regulated, the technology can be used on non-commodity broadacre crops because the regulatory costs are minimal. And this allows companies to be involved. Sorry, this allows small companies to be involved and not just the big companies like the big four, the Bayer, Corteva, Syngenta, BASF. So if it is not a broadacre crop, you can also probably more easily manage and control the marketing of your product, whether you limit it to just a local market or export it only to those countries where gene-edited crops are not regulated. So in the US, there are many small startup companies that are just getting into this gene editing space because it's not regulated in the US, and they can work on minor crops where they can control the distribution. Yeah, so they don't face risk with exports, for example. But of course, I think it's still prudent to still do your market analysis and make sure that you don't face a risk there. So on this slide here, I just showed some of the companies in this space in the US, and you will see in column 2 there, there are the big companies that we're familiar with, the BASF, Bayers, and so on. And then the third column there are some of the startups, or some of them are not so startups anymore because they're worth billions now. But compared to the big four, they're still small. But you can see from this slide, yes, there are many companies coming into this space, at least in the US, if not elsewhere. So if you use gene editing on commodity crops, which is traded like current GM commodity crops, then you could also have high regulatory costs. And this can impose the same hurdles as for our current GM crops. You can market these crops under identity preservation of costs, like calyx high oleic soybean, which I just mentioned. But that's not ideal because the extra costs and risks involved could be also high. So the regulatory piece, even for gene-edited crops, could be very important depending on what crops you go into. How many countries will continue to regulate gene editing in the same way as GM crops, or have exemptions or reduced regulatory requirements at the moment, still developing. But apart from the EU, which does not distinguish between GM or gene-edited crops, most countries, I would say, are looking at treating gene editing differently and either exempting some forms of gene editing techniques or have lesser regulatory requirements. So on this slide, you can see, on a global scale, those countries marked in green here, those are the countries where some types of gene editing techniques have been exempted from regulations. The countries marked in blue, they are countries where gene editing is definitely captured under GM regulations. And this includes, for example, New Zealand and South Africa actually just in the last couple of days came out and made this announcement that, yes, they're going to regulate gene edited crops. But for the majority of the world, things are still developing. So with regards to gene editing in Asia Pacific, let's see what's happening in our part of the world. So this slide shows the two extreme positions taken by various countries in Asia, globally. Yeah, on the right is the EU position with strict regulations of gene-edited crops, and on the left, the Americas, North and South America, where gene editing is either exempt from regulations or not strictly regulated. In Asia Pacific, the situation is still developing, with countries kind of following in between the two. But generally, they're opting for the rational part of exempting certain types of gene-edited crops or having lesser requirements for some techniques. With the next slide, I'll go through in a little bit more detail at what's happening in some of the countries in Asia Pacific. So starting with the left column, Australia, well, I was going to summarize the situation in Australia in a couple of sentences, but Osman has already done a treatise on it. So it's not necessary for me to go into that, except to say that, as Osman said, as techniques such as SDN1 techniques are not going to be regulated under OGTR, and in Fazens, they're also looking at not requiring pre-market assessment. In Japan, well, again, I was going to talk about Japan briefly, but Dr. Kasai has also given us a good summary about Japan. So again, no need for me to go into much about their situation there, except to say maybe, they have 3 agencies involved there, and each one differs slightly, but they also basically exempt SDN1 techniques. They look at SDN2 on a case-by-case basis, and with cisgenesis, yeah, I think they exempt it for things like microorganisms, but not yet for plants. The Philippines, I think just recently passed their legislation on gene-edited crops, where they will exempt SDN1 type technologies and SDN2 technologies with insertions of less than 20 base pairs, and also products of cisgenesis. Singapore is the same. They exempt SDN1 and products of cisgenesis. But I think they haven't proclaimed their regulations yet, but we understand that will be soon. Indonesia has not issued anything officially, but we understand they have put the recommendations to the relevant ministries to exclude SDN1 technologies and look at SDN2 on a case-by-case basis. So, these countries in Asia Pacific appear to be aligned in exempting certain GE techniques such as SDN1 techniques, have maybe slightly different approaches to SDN2 and cisgenic products. Okay, so in the middle column there, we have a group of countries where the regulation of gene editing products is still being hotly debated. But even here, there appears to be a trend towards exempting techniques such as SDN1. And these includes Taiwan, India, Vietnam, and Thailand. But as I said, there's nothing certain in these countries yet, still very much hotly debated. And finally, on the right-hand side, there are 2 countries, China and Korea. And we know currently in these countries, their regulations capture gene editing technologies as it would GM technologies. However, in Korea, there seems to be some move to introduce an amendment to their act, which would allow exempting SDN1 techniques. But this needs a legislative change, which is, as we know, always a challenge. In China, draft regulations have been agreed to and submitted to the ministerial level, and the Ministry of Agriculture and Rural Affairs is expected to release their proposal for public comments in the near future. But at the moment, we don't know, what they are going to do, whether they are going to regulate everything or not. So we think there will be at least less stringent data requirements for GE products, even if there are no exemptions. We're hopeful anyway. So earlier on, someone already alluded to regulatory harmonization. So regulatory harmonization is still crucial for commercialization of gene-edited products. Without regulatory harmonization, the agricultural industry faces the same trade issues that have been plaguing us for decades, since the first commercialization of GM crops. If gene-edited products are regulated like GM, like in the EU, then it will almost certainly kill gene editing technologies like it has basically done for GM technology. Fortunately, I think the majority of the world is coming to the rational approach and applying the principle that like products should be regulated in a similar way. Yeah, that is, if you can achieve the same result using either gene editing technologies or conventional or traditional seed breeding techniques, then these products should be treated similarly from a regulatory point of view and be exempt from regulations. Regulations really do need to reflect such an approach, so that the benefits of the technology can be accessed by all. And to my last point there, look, sometimes I think countries are holding out on freeing up gene editing technology regulations, not because they disagree with the science, that gene editing techniques are safe, but more because of concerns about pressure from anti-GN activists. And that I don't think is good for progress. So thank you very much for that.
Thank you very much, Dr. Keiku. I see that there's not much new and interesting questions in our Q&A and chat box. Let's give a round of applause to our two speakers. And before we go to the Q&A portion, let me ask staff to please post the post-webinar survey so that our audience can start working on it. I have a few questions. Can we have Dr. Osman also in the spotlight? When we were talking about the 25 years of GM crop adoption, and we see that there were good, policies on GM regulation is of course different from genome editing. So I was thinking that there are countries who have been planting biotech crops or GM crops would be more amenable to genome editing. Do you find it that way, or with the exception of Japan, for example? Can we have Osman, please, and then Dr. Keiku on that matter? Can we highlight spotlight Osman? There you go. Okay.
Thank you.
Thank you,
Ola. And look, I think I agree with your statement, because if you look at countries like the Philippines, which have a long history of cultivation of GM crops, they've recently made some very positive moves around the regulation of gene editing in what's been proposed there. If you look at countries such as North America and Canada, they also have considerable experience with the cultivation of GM crops, and of course South America. We haven't spoken about South America in today's webinar, but they've really been the leaders, the regulatory leaders when it comes to gene editing, and Argentina and Brazil have got decades of experience in cultivation of GM crops. So I think that comfort and that familiarity with gene technology has certainly led regulators in these countries to perhaps have a better understanding of the risk indicators when it comes to gene editing.
Yes, and your opinion on that, sir, Dr. Keiku?
Yes, I agree entirely with Osman. And one of the slides I showed you showed companies involved in this space in the US. A lot of these companies are in the US. So yes, where the opportunity presents itself, these companies will come online, and you see all the companies in the US and none in the EU.
Yeah, wonderful. Now, one of the things that I got interested in is in most of the countries which are preparing their genome editing genomic policy considerations, how flexible can their regulations be? Osman, you have mentioned about this, that since there are still new innovations coming up, what do you mean by flexibility, and how flexible should it be? And what are the other considerations that you have to put in there?
So I think it's really important that, well, let's use Australia as an example. So we have a process-based definition for gene technology. So if you have a product that you've used gene technology and it's captured by regulation, but your product is the same as conventional breeding, then, you know, there needs to be a quick exit point, and it doesn't matter about the technology you used. My argument is that you need to look at the risks in your final product. Are there any risks to human health and safety or the environment in your final product? And if you did it through conventional breeding, you know, use gene editing, then all we're saying that we know that conventional breeding has a history of safe use. So we don't feel in Australia that, you know, as long as it meets, you know, general food safety laws, there's not the need for pre-market assessment. Like for GMOs. And so this is where I just think there needs to be some broader thinking by our regulators to say, okay, where is the risk and what are we regulating and why? This is important. Why are we regulating it? Because it's, you know, it can be nice to regulate things. There's lots of nice to know, but I think regulators need to focus on what they need to know. And that's the identification of any risk to human health and safety or the environment.
Yeah, I find, thank you, Osman. There's one interesting question here from anonymous attendee. This is for Dr. Keku. With a variety of institutions, companies engaged in product development through commercialization of gene-edited products, please share your thoughts on government supporting or providing assistance programs, especially to medium-sized companies in navigating and securing authorizations in overseas market. I think this is when products are already ripe to go to commercialization.
I don't think governments provide a lot of support to companies to commercialize or to get approvals in various parts of the globe. We know, for example, the US government, for example, through their various USDA offices throughout the world, they do push for science-based proportionate regulations for these technologies. Yeah. So from that point of view, they support the technology, but they don't push for any specific companies. So like, for my company, for example, you know, we certainly don't get any subsidies or any direct support from any governments. We have to do it all under our own.
On our own.
Yeah.
Yes. Yeah. Wonderful. Good answer, sir. There's one here on, if you're familiar with the Nagoya Protocol, what is the role of Nagoya Protocol in considerations of commercializing gene-edited crops? Is it already touched in the Nagoya Protocol, the genome editing?
Sorry, I'm not familiar enough with Nagoya Protocol to answer this question, but perhaps Kay is.
I'm not sure which part of the Nagoya Protocol we're talking about, whether it's the Nagoya Protocol liability or the access and benefits part.
Okay.
So there are two parts of the Nagoya Protocol.
Aradna Sadasivam. Okay, she's not.
Yeah, well, you know, with regards to liability, I'm not sure that, you know, some of these products should be captured under the biosafety protocol or Cartagena protocol anyway. So they might lie outside of that. But if, for example, they are captured under the Cartagena protocol, then of course the Nagoya Protocol on liability applies to them. But if you're talking about the Nagoya Protocol on access and benefits sharing, then it depends on, of course, the source of your genetic resource. Yeah.
Okay. Wow, wonderful. I really have to cut short our discussion, and we had many very expert speakers today, and from many countries, from China, Japan, India, and Pakistan, and of course from you too. But we don't have so much time. I hope that our audience would be able to look at the post-webinar survey. I think there's a problem on that. But now at this point, I would like to give thanks to all of our speakers. And can we now call on Dr. Michael Jones to give the closing remarks, please?
Okay. Thank you very much, Ola. So this webinar is part of a series of joint activities between ISA and Murdoch University in Perth. And the aims of these seminars are to determine, look at the regulations and policies on gene editing in Southeast Asia and Australia. And you notice I always use the word gene editing. We've had this discussion, but I prefer gene editing because I think that implies a specificity, while a genome is less precise. Anyway, we also want to move the debate forward to see how we can best apply gene editing technologies to benefit growers, consumers, and help underpin future food security. And we really want to ensure that the issues surrounding GM technologies are not repeated for gene edited products. And I shouldn't go on. So first, I'd like to sincerely thank all our speakers for their timely and informative presentations. I think it's been really, really useful. And these have included specific country perspectives, broadening out to their application and the implications for policy and trade in gene edited products. And I hope that these presentations have helped all participants understand the importance of regulations, gene edited crops, and, as has been emphasized, the vital need for harmonization of regulations internationally. Because without this, all the promise and the beneficial contributions that this suite of exciting technologies can bring to agriculture will not be realized. But there are some great examples of how simple and clear regulations enable us to use these technologies. And in these talks, I'll highlight, for example, regulations in Japan as being a shining example in our region. And as has been mentioned, other countries such as Argentina are, in South America, at least 5 years ahead of the Asia Pacific in terms of policy enabling the use of gene editing technologies. And that's made a huge difference to the type of, smaller SMEs and government organizations are now being able to commercialize these technologies. And finally, I must thank the ISA team for arranging the webinar together with Adil and myself from Murdoch University, and also the Australian Government Project for Assisting Small Exporters for providing funding support for this webinar. And with that, I thank everyone who's stayed on for all this time. It's been actually a large number of people who've stayed through the whole event. Thank you very much. And I think it's now time to bring the webinar to a close. And so stay safe and thank you very much for attending.
Thank you, everyone.
Thank you, everyone.
Thanks, everyone.
Do contact ISA if you need information or if you have any collaborations. We are here to support the adoption approval of the new technologies in gene, and one of it is, of course, gene editing. Thank you, Murdoch University, Professor Mike Jones and Adil.
Thank you, ISA team, again, because I think you've been a great advocate for these technologies over the years, and I think you've been doing a great job.